Regulating eukaryotic gene expression. Eukaryotes are more complex than prokaryotes, so their methods of gene expression are more complex. These complexities include, not only are eukaryotes multicellular, there are multiple types of cells, each requiring a different regulation mechanism. DNA chromosomes are much larger and more complex than prokaryotic chromosomes. In addition, genes for similar functions can be found on different chromosomes and are harder to get to because of the double helix. In prokaryotes, transcription and translation occur in the same place, the cytoplasm. Transcription occurs in the eukaryote nucleus, and translation occurs in the eukaryote cytoplasm. This requires different regulation schemes. Cell differentiation. Every cell in a multicellular eukaryote contains the same genome. In other words, every cell in an organism has all the genes necessary to make all of its parts. But different genes are expressed for tissue cells and tissue-specific proteins, causing them to look and function differently, becoming more specific cells, or cell differentiation. Mathematically, cell differentiation can be thought of in terms of how many genes can be expressed over time. The percentage of genes in a cell's genome that it can express decreases as the cell matures. Transcription factors. The regulation of gene expression can be caused by groups of genes that can be influenced by the same transcription factors. Transcription factors are special proteins that either enable or prevent the transcription process from starting. Cell differentiation examples. Muscle cells, which you see on the left, and brain cells or neurons, which you see on the right, have the same DNA, but they're expressing different genes. That is why their structure and function are so different. In eukaryotes, gene expression can be controlled at different parts of the process. Chromosomal unpacking. Depending on how a chromosome unwinds, only selected genes are accessible to the transcription process. Genes that are tightly packed do not get expressed. Transcription. Special proteins called transcription factors either enable or prevent the transcription process from starting. mRNA processing. Some regions of mRNA get excised before it leaves the nucleus. Different patterns of excision lead to different proteins being synthesized. Translation. mRNA can be blocked from the ribosomes to prevent protein synthesis, and mRNA and unwanted proteins are recycled. Epigenetics. Epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Unlike genetic changes, epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reads a DNA sequence. Unlike prokaryotes, which only have one place to control gene expression, eukaryotes have multiple places where gene expression can be controlled. In order: 1. Packing and unpacking DNA, 2. Transcription, 3. mRNA processing, 4. mRNA transport occurs where the mRNA transits the nucleus cytoplasm boundary, 5. Translation, which occurs in the cytoplasm as a part of the translation process, protein processing, and mRNA and protein degradation. So we'll look at each one of these in detail in this order in the slides to come. The first place that this regulation can occur is in packing and unpacking DNA. In order to fit DNA into the nucleus, it's coiled and folded in several steps from the double helix until a chromosome. When DNA is packed in chromatin, it is not accessible to RNA polymerase, so transcription cannot happen. The main factor in the specialization of cells in multicellular organisms is the control of which genes are unpacked from the chromatin to be exposed to RNA polymerase. So in this double helix, all gene sequences are exposed to RNA polymerase. The next sort of order that the DNA has is wrapped around histone octamers to form nucleosomes. In that case, some genes are exposed, and when the nucleosomes are compacted into a chromatin fiber, no genes are exposed and transcription cannot happen. Genes that are tightly packed do not get synthesized. DNA must be unwound before transcription starts. Genes that are unnecessary for a particular cell will remain packed while the necessary ones are unpacked. Unwound DNA, or euchromatin, is loosely packed DNA and can be transcribed. You can see it as these white on the photo. Heterochromatin is tightly packed and is not transcribed. You can see that as these tightly packed little black spots. Shown is a human cell nucleus with these two different types of DNA. Adding methyl groups, known as methylation, to DNA blocks the transcription factors from attaching, preventing transcription. The methyl groups do not change the DNA sequence and are known as negative regulatory molecules. They typically cause an almost permanent inactivation of a gene. So here are our unmodified bases. You can see adenine, cytosine. The methyl groups only attach to adenine and cytosine. So you can see the methyl group here on the adenine, or there's two locations where it can modify the cytosine, here or here. Methylation allows the differentiation of cells during embryonic development. It turns off genes not needed by a particular type of cell, which is why different cells have unique functions. Methylation is an epigenetic modification. In acetylation, the addition of an acetyl group, COCH3, so it's carbon double bond O, carbon single bond CH3, loosens the histone's attachment to the DNA molecule, enabling transcription and turning on the genes. So we have acetylation, so we're binding these acetyl groups, and you can see that that's helping to unwind the genes. Deacetylation would then remove those acetyl groups, causing the DNA to be tightly wound again, and then that would turn the gene off. 2, transcription. This is the second place that gene regulation can occur, and that's at transcription. Special proteins called transcription factors either enable or prevent RNA polymerase from attaching to the promoter region. So you can see we have the RNA polymerase in the promoter region and several transcription factors. The promoter region is upstream and adjacent to the gene being transcribed. So you can see gene A here, the gene being transcribed. When the transcription factors attach to enhancers, that can be thousands of nucleotides away from the gene to be transcribed. The factors are called activators. So you can see those here. Here's an enhancer, and here are our activators. Because of the distance between the activators here and the gene to be expressed here, the DNA molecule must fold up to place the activator next to the promoter and the RNA polymerase. So this fold happens so that this all is in proximity. Factors that prevent the attachment of RNA polymerase are called repressors and can be attached either to the promoter or enhancer region. The third place that we can get gene expression regulation in a eukaryotic cell is in mRNA processing. RNA is spliced in different ways, allowing a great number of different mRNA molecules and then proteins to be created from the same gene. The mechanics of this process was covered in the RNA processing topic in the central dogma unit. So you can see that we have different exons that can be spliced in different ways. So this mRNA has all 5 exons. This only has 1, 2, 4, and 5. This has 1, 2, 3, and 5. And that alternative splicing translates to 3 different proteins, A, B, and C. 4. mRNA transport. The nuclear pore is a protein structure within the nuclear membrane. Each nuclear pore, shown in the sketch below, is made up of hundreds of individual proteins that ensure only mRNA molecules with proper caps and tails can make it to the cytoplasm. This control bridges transcription and translation. 5. Translation. Short segments of RNA called interfering RNAs, iRNA, prevent translation from mRNA to protein by binding to mRNA molecules. They create sections of double-stranded mRNA, which identifies them for destruction in the cytoplasm. This occurs in the cytoplasm and the nucleus, where they prevent transcription by binding to the chromatin. 6. Protein processing. Regulatory proteins attach to the 5' end of mRNA. This prevents the attachment of ribosomal subunits and tRNA, blocking the translation of mRNA to proteins. 7. Protein degradation. Proteins don't last forever. As they age, their shape changes and their functionality decreases until they don't function at all. They're labeled with a polypeptide, ubiquitin, that labels them for destruction. They're broken down into amino acids by proteasomes. The amino acids are recycled. Degradation of mRNA. Hydrolytic enzymes in the cytoplasm break down mRNA molecules. The length of time an mRNA survives in the cytoplasm relates to how much protein is made from it. Longer time in the cytoplasm means more translation by ribosomes. mRNA can last for hours to weeks in the cytoplasm. The length of the poly-A tail is one of many factors that determines the time of survival in cytoplasm. The longer the tail, the longer its survival. mRNA gets degraded because it's an unstable molecule, and iRNA molecules tag it for destruction. Hydrolytic enzymes break down molecules into their components in the presence of water. Here's a summary of gene expression regulation in eukaryotes. The gene is unpacked from chromatin. The right transcription factors must be present, and mRNA is formed. So that's how transcription occurs. The cap and tail are added to the mRNA. The pre-mRNA is edited or spliced and becomes mRNA. Nuclear pores allow mRNA passage to the cytoplasm. mRNA comes into contact with a ribosome and begins the protein formation. So that's the process of translation. And proteins are made and used within the cell or exported to the environment.